we can.
Okay, we can go ahead and start. Hello, everyone. Thank you for coming to our event. Welcome to our Inspiring Scientists event with Dr. Balasubramanian. This event, again, is hosted by CCNY Women in Science. We're an organization at the City College of New York, and we aim to increase awareness about the status of women in science, as well as promote inclusive and a really healthy environment within the scientific community. So throughout the academic year, we do host several events to highlight people in science, not only to showcase their work, but also to allow us as students, as faculty, to really just ask questions about their lifestyle and their journey through science. So today we have the lovely Dr. Balasubramanian, who is an associate research scientist in the Department of Ophthalmology at Columbia University. Dr. Balasubramanian completed her PhD in neurobiology at the University of Rochester, where she studied retinal development. She then furthered her research as a postdoc at Columbia University, where she delved into researching about cell signaling in early mammalian eye development and its impact on many pediatric eye diseases. In addition to this, she is also an active member of the Columbia University Postdoctoral Society and advocates for women, immigrants, and people of color in science and hopes to make science a more equitable ground for parents and especially mothers. So before we start, I just want to mention that you guys are allowed to ask questions throughout the presentation. Our Q&A session will be hosted by Marilyn, and I think Maduna will chip in for a few questions as well. But you guys are also welcome to ask questions and type your questions within the chat throughout the presentation. Or you can unmute during our Q&A session. So without further ado, Dr. Balasubramanian, you are welcome to start whenever you're ready. Do you have permission to share your screen?
Yes.
Okay,
thank you so much. I'm going to go ahead and share my screen now. Can you all see my presentation? Yes.
Yes, thank you.
Thank you, Sacha. Thank you for the very wonderful introduction. I'm currently an associate research scientist in the Department of Ophthalmology at Columbia University. And I just want to give you all a brief, window into what my career path as a vision scientist has been so far. And hopefully, what you will take away from this is that, you can follow many different paths, and everybody's path is unique and everybody's path is their own. And as long as you kind of, keep at it and know what you, want to take out of your career path, that's all that matters at the end of it. So hope I can, give you a brief glimpse into it, and also, hopefully inspire you to stay true and stay true to your path, whatever that might be. Okay. So I am a vision scientist, and I really did not start out as a vision scientist. I'm originally from India, and I have my bachelor's in engineering, actually. And there is a saying in India, which is, you either become an engineer or a doctor, and then you figure out what you want to do with your life. So that I followed, true to that, and I became an engineer. And that's when I realized that I did not like anything to do with engineering, and I actually wanted to be, a biologist. So I came here to the United States and did my master's in neurobiology. And as part of my master's in neurobiology, I took this very nice course on cellular neuroscience, which had a huge genetic component focusing on vision genetics and vision biology. And that's when I realized that my true interest lies in understanding more about the biology of vision. And that's kind of where I started. So I enrolled in a PhD program at the University of Rochester right after having taken that course. And thankfully I got accepted because, I had no background in biology at all. And when I was accepted into the program, I started working in the lab of Dr. Lingan, who at the time was studying who was trying to understand the role of different transcription factors, especially limb domain transcription factors, and their role in retinal development. So I spent my 5 years of PhD studying retinal development at the University of Rochester. And also as part of my PhD, I did quite a bit of studies on injury and regeneration in the eye, looking at several different cells and cell types within the eye and the retina, that can be programmed towards regeneration. And then I moved here to Columbia for my postdoctoral work, and I continued studying development of the eye, but with a disease relevance in early, sort of pediatric and congenital diseases such as aniridia and congenital glaucoma. So I continued my work here in Columbia. As part of it, I also did some disease modeling of various eye-related structures using organoids. I'll talk about that a little bit. And now I'm using all of this expertise that I have, which I really did not have right off of my bachelor's, into studying disease variability in glaucoma as an associate research scientist in Columbia. So I won't spend a lot of time talking about my PhD work on development and injury and regeneration. I'll give you a brief insight or brief glimpse into some of the development and disease work that I did as part of my postdoctoral career, and some of the work that I'm currently doing using genomics. So I'll just briefly mention that my development work sought to understand the role of limb homeodomain transcription factors, especially a transcription factor called as LHX9, and its role in the differentiation of different amacrine cell subtypes. As part of this, I also, like I mentioned, did some injury and regeneration work, for which I got the NISTEM predoctoral grant. Which was to understand the stem cell-like characteristics of Mueller glial cells, which are a particular kind of cells within the retina, and their ability to promote retinal ganglion cell regeneration, or their ability to be reprogrammed towards regeneration. The bulk of my time I'm going to spend is just talking about this, which is a publication that I recently authored, which is studying the role of FGF signaling, which is a growth factor, which is called the fibroblast growth factor signaling in the development of peripheral retina, and how it disrupts the development of the ciliary margin and causes aniridia. So I'll go ahead and get started with just talking very, very briefly about this to give you a glimpse into what this work is about. So the ciliary margin itself is structure at the very tip of the eye, and what that means is basically during the development of this optic cup, which differentiates into inner neural retina and the retinal pigment epithelium. There's a structure at the junction of the neural retina and the retinal pigment epithelium, which gives rise to different structures like the iris, the ciliary body, and several anterior segment structures. The iris, determines the color of the eye, but apart from the vanity aspect of it, it also determines, your pupillary constriction, and there are several biological functions associated with it. And also the ciliary body gives rise to, produces aqueous humor, which maintains the intraocular pressure in your eye. So it's very important that we understand where these structures are coming from and what the ciliary margin, which is at the junction of the neural retina and the retinal pigment epithelium, is actually doing. So why do we care? Again, we need to understand the normal development of the ciliary margin, because a misspecification of this leads to many congenital forms of vision loss, such as congenital glaucoma, aniridia, where there is a clear lack of iris, or coloboma, where there's like a teardrop iris formation. And these all have some kinds of vision deficits, and it's really important that we understand how this comes about to be. And to do that, we need to understand how the normal development happens. So again, like I mentioned, this is a cross-section of an eye, of a developing mouse eye, at an embryonic stage, at a pretty early embryonic stage, where you can kind of clearly see the presence of the neural retina and the retinal pigment epithelial layer here in the back, the optic nerve coming off of the retina, the lens, and the surface ectoderm, or like these cells at the top of the eye, which gave rise to the lens prior to this. And then this here is the ciliary margin, which is sort of this innocuous little structure at the junction of the retina and the retinal pigment epithelium. What I identified as part of my postdoctoral work was that FGF signaling, or fibroblast growth factor signaling, is very high in the neural retina, and there is a complementary expression of Wnt signaling in the ciliary margin and the retinal pigment epithelium. So if you, I guess you can appreciate that the expression of FGF and Wnt signaling is very compartmentalized, with the FGF being really high in the neural retina, while that of the Wnt signaling being really high in the ciliary margin. So we were trying to understand what is the how do FGF and Wnt signaling. Sure. Did somebody have a question? Okay, sorry, I thought I heard something.
Sorry, I think they might have had a question, but if you do have a question, you're welcome to put it in the chat, and then we can get back to it at the end of the presentation. Thank you.
So what we were trying to understand was how these two signaling moieties interact. How does FGF interact with Wnt, and how does Wnt interact with FGF in determining how the fate acquisition or how the ciliary margin comes about to be, and what actually goes wrong, or what happens when we have like various congenital diseases like such as aniridia. To cut a very, very, very long story short, what we found was that there was an interaction between different kinds of FGF, ligands and signaling within the neural retina, which is here, and that of Wnt signaling, which is arising from the retinal pigment epithelium, and they kind of come together, and they, not just, they don't just have a complementary expression, but they actually interact with each other at the ciliary margin. And that FGF signaling is actually needed for the ciliary margin to develop, and for the Wnt signaling to be to come about to be within the ciliary margin itself. So we showed that this interaction happens using in vivo mouse models, in vitro studies using organoids, and also some single cell genomics to understand what this interaction might be. So again, this is more, going into the details of what the study actually was, where we genetically ablated components of the FGF signaling, and saw that the ciliary margin was sort of it expanded. So at the when there is a loss of FGF signaling, there's a lot more ciliary margin, or the tip of the retina, kind of starts to expand into the neural retinal space at the expense of the neural retina when compared to the control. So if we lose FGF signaling, we're just getting so much more of the ciliary margin. And it also leads to aniridia, where the mouse eyes look like they just have this open sort of iris phenotype. So all of this kind of blends in well with what we understand about the role of the ciliary margin, which is that they are in fact needed for the development of structures such as the iris and the ciliary body, and how FGF signaling affects maintenance of this neurogenic boundary, or maintenance of this boundary between the neural retina and the ciliary margin. And that, the ciliary margin is really, for a long time, was thought to be very similar to that of the neural retina. But now we understand that it's not, that the ciliary margin itself is, a different structure compared to that of the neural retina. Again, a lot of mouse studies that I'm sort of summarizing over here, we genetically ablated different FGF ligands, such as FGF3, 9, and 15. And what we saw was a gradual reduction, or a gradual expansion of the ciliary margin. The more number of ligands we ablated, the more number of ligands we removed from the ciliary margin in using mouse models, the larger the expansion of the ciliary margin became. So this gave us an idea that the ciliary margin is not just dependent on FGF signaling, but it's also dose dependent, meaning that depending on the amount of FGF signaling that actually goes in, that the eye is exposed to, the ciliary margin is, kind of develops accordingly. I also incorporated some single cell genomics into my study to understand how the different aspects of the ciliary margin, basically the distal, medial, and the proximal aspects of the ciliary margin come about to be. Because we know that this, now we know that the ciliary margin is not just an extension of the neural retina. We know that it's its own structure. There is also, differences within the same structure. There are different aspects to that structure. There are different cells that, behave differently within the ciliary margin itself. And we were able to resolve the differences in the ciliary margin itself using some single cell genomics. And I also, using single cell genomics, we were also able to show that FGF signaling not only is needed to keep a lot of progenitor cells or the developing cells cycling, but also it's needed for the differentiation of the ciliary margin itself, meaning in the absence of FGF signaling, these progenitors tend to leave their cell cycling. As you see in the control here, there are a lot of progenitors cycling within, but in the absence of FGF signaling, they tend to leave the cell cycle and exit more towards the ciliary marginal fate. And again, using iPSC organoids, what we showed was that if we that there is a dose-dependent effect, in fact, and that depending on the amount of FGF or Wnt signaling we actually expose the optic cup to, we get different amounts of the ciliary margin. Marked here in red is the retinal pigment epithelium, and green is the neural retina, and in yellow here in between is the ciliary margin. And I think you can appreciate that there is a lot of ciliary margin here when we modulate the response, or when we modulate how much FGF and Wnt goes in towards the developing optic cup in an organoid model. So some of the outcomes of this research, which was recently published, was that we now have a new target for genetic screening in patients with aniridia, and that these, we were actually find a developmental basis for aniridia, which was previously known to be a single gene disease, which affected just by PAC6. And we know that we can tie the developmental basis of aniridia to aberrations in the ciliary margin. We're also able to distinguish this, the neurogenic boundary between the ciliary margin and the neural retina itself. And it opens up potentials for early life intervention and gene therapies to actually treat aniridia early in life. And I briefly spoke about the disease modeling aspect of my career, which right now what we are trying to understand is, can we make organoids from mouse embryonic stem cells to actually model aniridia? So we can actually study that in a dish. And this is some of the work that's been ongoing in the lab after the conclusion of my study. So we're trying to model aniridia to see if we can actually understand the early basis and what actually happens even before the ciliary margin is specified in these organoids. So I spoke about the development and disease aspect of it. I'll just spend a little bit of time talking about the disease variability and glaucoma aspect, which is what I'm currently working on. So again, like I mentioned, throughout the course of my career, I've worked with mice. I've worked with, I've done my fair share of in vivo studies, studying development. I've recently started to incorporate iPSCs or embryonic stem cell cultures into my study as an in vitro aspect of it. And I've also used some single cell genomics to study how the development comes about to be. These were all like very recent techniques that I had a lot of hesitancy in trying to incorporate all of these because, there's always the fear of the unknown. But it has brought me into this foray of single cell genomics and trying to understand disease variability. So what are we trying to understand in terms of disease variability? Glaucoma is a disease of the eye that affects about 80 million people worldwide, and glaucoma comes in many various forms. There can be congenital forms of glaucoma, early onset glaucoma, late onset glaucoma, glaucoma without any intraocular pressure elevations, and so on. So there are very many flavors of glaucoma, and it also affects, people of color differently than it affects Caucasians, or it affects Asians, and so on. So there is a very specific race effect in how glaucoma affects people. So we know that the background, there is a difference in background can affect your susceptibility, variability, and the degree of disease progression with glaucoma. So we wanted to try and understand what actually brings about this variability. Like, why do we have this variability, and why do we care? What we wanted to understand was the front of the eye, coming back to, again, the structures, the ciliary body and the iris, which I spent a lot of time studying the development of. I wanted to pivot and now study the how it actually causes disease. The aqueous humor that's produced by the ciliary body in the eye is what makes the eye buoyant and keeps the intraocular pressure intact, and any variations to the intraocular pressure contributes to glaucoma, or leads to glaucoma. Glaucoma essentially, means the endpoint of it means that there is a severe, there's certain degree of optic nerve damage, or the loss of retinal ganglion cells. But it arises from a higher intraocular pressure. The higher intraocular pressure is a very, very known and a major risk factor for glaucoma, and so is age. So we wanted to see what are the differences in how the intraocular pressure is regulated by the ciliary body and the iris in various different backgrounds and genetic mutations. So we honed in on one gene, which is called the LMX1B, which has been identified in human Jiva studies as a key relevant gene, and has been associated with high intraocular pressure and with pediatric glaucoma or congenital glaucoma. What we noticed was a particular mutation in LMX1B, where at this locus, at the 265 locus, actually led to a loss of the structures of the Schlemm's canal and the trabecular meshwork, which are these key aqueous humor drainage structures, which is what recycles the aqueous humor drainage from the front of the eye in towards the veins, and then the ciliary body produces more aqueous humor and then gets drained out again. So without these drainage structures, the aqueous humor sort of builds up in the anterior portion of the eye, leading to higher intraocular pressure. And what we found was that this genetic mutation actually causes an aberrant drainage structure. So there is no place for the aqueous humor to drain, and leads to increased intraocular pressure and also glaucoma. So what I'm trying to understand now is we have these very many mutations of glaucoma, such as the Q105X, V265D, there's a splice mutation of C86X, and many other mutations that we have identified with forward genetic screens at the LMX1B locus. And we're trying to identify how these different mutations develop glaucoma. Like, for instance, some of these mutations develop glaucoma at a later stage, so we have a late onset glaucoma. Some of these develop at an earlier stage, and so it's an early stage of glaucoma. And some of these do not have glaucoma, and some of these have a very structural sort of abnormality in glaucoma, and so on. So using this particular gene as a proxy for the genetic background, we're trying to understand how different mutations have this variability in disease. And we're also trying to understand if there are different structural changes to the aqueous humor drainage structures, such as the Schlemm's canal and trabecular meshwork in these LMX1B variants. So I'm doing that in the aspect of disease, and also trying to understand the mechanism by which this happens. And the way we are approaching that is to see if the Schlemm's canal and trabecular meshwork cells identified using single cell genomics undergo these molecular or transcriptomic changes deep within their within their transcriptome itself, and if these changes can be attributed to whether they acquire, whether the disease has a early onset or a late onset phenotype, or a lesser degree of severity, and so on. So that's one thing that one of the parts of the project that I'm still working on. And the last aspect of it is to develop treatment modalities. And so we're working with AAVs, or adeno-associated viral vectors, to actually specifically see if we can target just these aqueous humor drainage structures and rescue or add particular genes, or maybe even delete genes in an effort to correct these either early or late onset types of glaucoma, or even not very severe types of glaucoma, in an effort to rescue the phenotype, or to even prevent some of the phenotypes from being acquired later in the stages. So that kind of brings me to where I am at right now. A lot of this work is still in progress, and I don't want to bore you with a lot of the details. So I just wanted to give you a very, very, broad level overview of where how my career just came about to be as a vision scientist, from starting out to being an engineer in India. And I hope I've convinced you that, acquiring new, either be it techniques, or either be it a new field of study altogether, is not it's completely doable. And yeah, so this is where I'm at right now. So with that, I'll end my talk by saying thank you to all of my faculty collaborators and mentors I've had throughout the years. No work ever gets done alone. And of course, apart from all of these, all the wonderful colleagues I've had, including Miruna here, who has been, who's been such a strong support system for me. And also, of course, all of my funding sources. And like a very huge thanks, especially to all the mice that go into the study, because none of this would have been possible without a deep appreciation for how we use mice in our studies and what we can learn from them. So with that, I'll say thank you, and I'll stop sharing my screen, and I'm happy to take questions.
Thank you so much, Dr. Balasubramanian, for showing us an overview of your work and your research, and sharing a little bit of your whole life story with us. So before I start out with questions, which it will be more of a free-flowing conversation, and we'll discuss any topic that comes up, would anyone like to ask any questions first about her work, or anything specific about her life? I'll open it up to everybody right now for a few minutes.
Can I ask a very simple, a small technical question, which I honestly don't know the answer to? What is the dynamics of the blood vessel development at early stages? Do they play a role? Is there like an immune component of it?
The answer to all of that is yes. So yes, there is a huge immune component to the development of the anterior segment, or the anterior structures of the eye. And the development of blood vessels is very intrinsically intertwined with that of the development of the Schlemm's canal, the trabecular meshwork, and a lot of the anterior segment. Actually, Schlemm's canal kind of branches off of the blood vessel development. It's actually, it has its own term called the canalogenesis, but it actually comes off of angiogenesis and lymphangiogenesis. A major part of my work now is trying to understand whether Schlemm's canal resembles the blood vessels more, or the lymphatics more, or if they have a unique signature of their own, which they do, but the developmental origins do trace back to vasculogenesis. And also there is a huge immune component to the development of these aqueous humor drainage structures. And that's also something that we're trying to understand is, what are the role of macrophages, for instance, and how do they, modulate and, modify these various developmental activities, for sure. Yeah.
Thank you. Makes sense. Yeah, so like Marilyn said, like I think from now we can just like open it to a very, this is something I really enjoy about these sessions, is that it's like really having a cup of tea with some friends and people that you know in science and like learn from them. And so please everyone participate as much as you want. Marilyn, do you want to start with questions, or everyone feel free to do so?
I have a question. Can I ask?
Yeah,
Sure.
go for it. Go for it.
Okay, so I was just like curious, like when you are a retina biologist, would you say like it's kind of easy to go back and forth from like doing development and then disease? Like if I, let's say, wanted to, like, I'm studying development, but if later on I wanted to study retinal diseases, like would I kind of be like, is this a good, how do those like interchange in this
Absolutely.
field?
I think the answer to that is I think you can do whatever you set your mind to and whatever you want. I had the same consternation initially when I started studying disease, because that was my overall goal, is to apply what I learn in development towards understanding disease later on in my career. But when I actually set foot into disease, I was very hesitant because I didn't know anything about disease. I didn't know anything about oh my God, like there is just so much variability in my genetic mutants during development. Everything looks the same. I have an, N of 3 or 4, but in disease, suddenly your N's become like 40, 50, like, you have to have so many mice and so many experiments and so many things that you have to think about when you're thinking about disease, because no disease is actually alike. And then if you have to think about translating that into humans, forget it. Like there is no, no one disease actually looks the same between humans. But what it gave me an appreciation for is how to apply what I learned into, what will actually be useful, and kind of how can we translate what we learn into, into studies into the human. So that being said, I did have my fair share of trouble understanding and, kind of wrapping my head around the complexity of disease. But I truly feel like if you have your PhD studying development, I think you can study disease or development and disease, or, anything else for that matter. And it shouldn't take you pretty much any time to, grasp and understand the concepts and kind of build your science around it. Because at the end of the day, I think science is science. I mean, and once you understand the basics behind it and the background of what you're dealing with and the complexity of what you're dealing with, you can frame your questions around it.
Exactly. Thank you so much. That was a very, very poignant answer. Thank you.
I have to say Revathi is the best person to answer this question because she masters both development and disease. So
yeah, absolutely.
Yes, 100%.
I don't know if mastering is the right word. I'm a jack, if anything, of all, sort of trades at this point. But yeah, I mean, I do have a very deep appreciation for the basic science that goes into development. And now I also have a very good appreciation for the translational science that goes into disease. So they're two different beasts, but at the same time, there is a lot you can take from development and apply to disease and vice versa.
Yeah, so I actually have a little bit more of a general question, not specifically about the research, but just about transitioning from being an engineer into biology. So I'm an undergrad student, and I did not start undergrad with the career path that I'm going into right now. So how was it for you transitioning from engineering to biology, and did your community and your family accept that transition, or was it difficult for you?
I mean, like I said, you're either an engineer or a doctor, and then you figure out your life. Because I was an engineer, my parents didn't care, and my family was very supportive. They were like, okay, you got it. So, but I have to say, when I started my PhD, it was very, very difficult transitioning. I did not know how to think in biology terms. I didn't have a lot of research. I didn't have any research experience. I hadn't worked in a lab. So, in light of all this, I don't know how I got accepted into a grad program. But when I did, and I ended up in Lynn's lab after my first rotation, we still joke about it. I have, whenever I talk to Lynn, he keeps joking that after my first rotation, he sat me down and he said, maybe biology is not your thing. And he was very nice about it. And he said, it's going to be a huge learning curve for you. I'm happy for you to stay on in my lab, but it's going to be a very, very huge learning curve for you. So maybe why don't you explore some of the neuroengineering labs in the program instead of a basic biology lab? And that's kind of where I started. I still remember going home like completely and utterly devastated that day. Hearing that maybe biology wasn't it for me. But I persevered on. I came back and I told him that this is what I want to do, and this is truly what I'm interested in, and I'll do better, and I'll try better, and I'll try harder. And and I'm really glad that somebody gave me the chance, and Lynn took the chance on me and let me stay on in the lab and learn and fail again and fail some more. But I think it worked out eventually, and I'm glad, and I appreciate the privilege with which I'm saying that I can look back on it and joke about it. But I also understand not everybody gets that opportunity to do that. It was. The short answer to your question is it was rough. It was rough, not because of, I had a lot of community support, and I had a lot of support from my mentors, which, at that time I didn't appreciate as much. But it was rough. It was, the transition was something else. But again, I would say, if you are switching backgrounds, or if you're switching from something into, into something that you have not worked on previously or have no research experience previously, find yourself that supportive community or supportive set of mentors. That mentor need not even be your direct supervisor, it can be anybody else. It can be anybody in science. It can be whoever it is that you truly trust and respect, and find that person. And, hopefully you'll find that the support is enough to let you persevere and get to where you have to.
I have another question kind of to bounce off of Marilyn's question about engineering. I wanted to know, in any way, did your background in engineering help while in your research, whether it was like a concept or maybe like a technicality or techniques or anything? Did it help in any way?
Absolutely. For some reason, everybody just came up to me and asked me to fix all their equipment. And I didn't know how to. I really didn't. So the assumption that I could help my credibility in the lab, and I just figured out how to fix something. But yes, it did help. Having a background in engineering also gives you the mentality, or the you start to think in a particular way, and that is never a bad thing. You can apply the same thing to science, to research, to pretty much attacking a problem, trying to solve a problem, and so on. Where it did come in handy was all of my computational background came in handy when now I had to incorporate a lot of genomics work. I could dig back into some of the computational background that I had during my engineering, even though it was a decade ago. But there's a way of things just coming back to you, and certain things like just came back to me much easier, and I could reflect back on what I learned during my undergrad years, like in my computer science class, and sort of harness that into a lot of my genomic work that I've been doing now. So yes, it comes in handy. You never know when things come in handy, and also in repairing a lot of centrifuges, I think. So yeah.
I would also like to ask, sorry, Marilyn, go ahead.
If you can go ahead, I'll ask it.
Okay, I was asking, my question is definitely for all of us. So you mentioned something that I also find very useful, that your mentors are not necessarily your supervisors, and your mentors can be your friends.
Yes.
I think it's very difficult for a trainee entry level, and obviously the more you learn and the more you know how people react, the better you're at it. But what advice would you have for entry level people to kind of decide that, and how to do it without upsetting your supervisor or without stepping on anyone's toes?
Absolutely. I think, it's a very tricky question, and it's a very tricky situation, because as a trainee who's just starting out, you don't know, who you can reach out to, and what your mentor would think. I mean, what your supervisor would think if you reached out to others, and so on. But what I've found generally is that ultimately it is your life and your career. Um, and almost everybody have encountered have always understood that, maybe the wavelength doesn't match, and maybe you need to talk to somebody else, and maybe you just needed to discuss some other aspect of your career. So it is a very acceptable thing to reach out to people and say, hey, I just needed, some advice on this, and then to build your relationship from there. And I will say this, because I mean there's I've all of my mentors and my supervisors know this, and I've always had a mentor who is not my supervisor. It's also because you can go to your mentor about things that you cannot discuss with your supervisor, such as, I really, really think I need to defend because my husband's in New York, I'm in Rochester, and I don't want to be in a long distance relationship anymore. Things like this I cannot take to my supervisor, but I can take to my mentor. And there are ways that you can, discuss and, come to a conclusion, or you can try and understand your situation a little bit better. However, that being said, it did not come easy. I think I just went with the motto of what's the worst thing they can say, which is a no. Hey, I just needed to talk to you about this, situation I had. I was wondering if I can pick your brain. The worst thing they can say is no. And that's okay. And if they don't have the time for it, and I've had my fair share of people say, I really don't have the bandwidth for this. I'm sorry, I wish I had more time. And then you say, thank you for your candor. I'm glad that you responded and said that you didn't have the time for it, and you look for somebody else. But I've always gone with the motto of, never hurts to ask, never hurts to ask. So, reach out to people. I think, if think about it in terms of, what would you say if somebody came and asked you, hey, I need help with this? What would you say? You would say, sure, let's talk, or no, sorry, I don't have the time. Or you would say, sure, but I don't know if I can help. Maybe I can point you to somebody who I know would be more able to help. And these are the three pretty much big answers that come to my mind. And so I just go with that and think, what if somebody came up to me for help, and just kind of self answer that question and just write that email and hit send, and then hope that they say yes. But I understand, I really do, I understand that it's really hard as a trainee to do that. But I think the further you go in your career, the more and more and more important it becomes that you have a mentor who is not your direct supervisor. It also helps, expanding your range of people who will be able to write you letters, who will be able to vouch for you, who will be able to say, I have this connection and I know this person, and I know this person is trying to do this, and set up a collaboration, for instance. So it just basically expands your social range, it expands your collaborative range, it expands your science range. There is no downside to reaching out to people. And as far as your supervisor goes, if they don't understand that you need other mentors in your career or for your personal growth, look for another supervisor. Maybe. But I think, every supervisor I have encountered has understood that they cannot offer everything. And when when I'm mentoring my RAs who work with me in the lab, I always say the same thing: there are certain things that I cannot help with, and that you should find somebody else to talk to, and you should find somebody else who can mentor you on all of this. And that is not something that I would be offended by.
Thank you.
Yeah, I'm glad you asked that, Marina, because my question was also in the same similar topic. But going off of that, with your RAs and your mentees in general, is there one big piece of advice that you always give them, generally, whether it's in terms of just facing obstacles or challenges that might come their way, or pursuing opportunities at all?
Yeah, I always, I mean, again, it depends on my mentee, but one common theme that I always tell them is to, again, never have just one mentor. Because there are always different things you can get from different mentors. And also the other thing that I say that I just covered is it never hurts to ask. It never does. I think we all get too much in our own heads when we want to draft that email and hit send and worry like to the moon and back about what the response might be. But it just never hurts to ask, whether be it a question, whether be it, for a collaboration, whether be it, for help, whether be it for, any kind of guidance, for anything at all. It just never hurts to ask.
Thank you. Yeah, so I also wanted to ask generally, because with WINS Women in Science, we focus a lot on discussing inequality and obstacles that we face as minorities, not just women, but in terms of race, religion, the LGBTQ community. There's a lot of things in the scientific field where we are often discriminated or looked upon differently. So were there any times or instances, or maybe not even your own personal experience, or just instances that you've witnessed where you've seen discrimination at your workplace, and how have you dealt with that?
Absolutely. I think as at some point as a woman, as a woman of color, as a immigrant, as a mother, as a parent, I have faced some sort of a passive discrimination, if not an active form of discrimination, where there is that, passing comment on, oh, why do you have to head out at 5 every day? Because I have to go pick up my child. Or, there is some passing comment or one, at every point or the other that I have faced, which has made me feel that there is, which, kind of stokes this imposter syndrome and the insecurities that you already deal with as, a minority or a woman in science, which is vastly underrepresented. But what I have, used as an arsenal is to have as many women in higher positions, be it the level of professors or provost or whatever position they are in, as my mentors, to seek them out, because I know that a lot of women and women of color or immigrants in these positions do understand where we come from and understand our challenges. It is hard to find women higher up in the administrative ladder, or in a lot of the higher professorial or chair of a department sort of positions. And that's where I think institutional commitment to diversity comes in a lot, and it comes in handy, because I can relate and I can talk to and I can have these different mentors, and they can understand and relate to me better and understand all of these challenges that I'm going through. But yes, I mean, who at every point there has been a particular a snide remark or a comment or a statement that has been thrown my way about either somebody recently told me, oh, you're really good at coding for a girl. And I and I was like, I don't know what to say to you, but okay. So so there are these kinds of comments that that really make your that start to affect your sense of ability. That start to affect your sense of when you're already insecure about certain things, it stokes this insecurity and the imposter syndrome that you tend to have as a woman more than anything else. Yeah, so I don't know if I really answered that question, but because I still kind of battle with it and I still deal with it on a daily basis. But what I've found best is to ignore things as best as possible for the things that don't matter, but to fight back as hard as you can for the things that do matter. Fight back for equity, fight back for, uh, for like equal chances at everything, fight back for that position at that table, and fight back for that chance to do something irrespective of whether you're a woman or a parent, or that I have to go pick up my daughter at 5 p.m. So yeah, I really don't know how else to answer your question, because like I said, I still struggle with it on a daily basis.
Yeah, I agree. I think it's something that we can never truly escape, especially in those little details. Because right now, it's not like every day you see such severe types of discrimination, but it's always very subtle nowadays, and it's very frequent. So yeah, it is something we deal with every day on all levels of whichever scientific field we're in. I also wanted to ask, because you're saying you're a mother, you're also, as we can tell, an accomplished researcher. So how are you able to balance your personal life and just being a mother with all your hard work and research? And is there ever a time where you just have to say no to work and then just head to family, or has it ever, been a problem for you?
Yes and no. I would say my priorities. So acknowledging that my priorities have shifted was a huge part of the solution. I initially, when I had my daughter, I could not accept for a large part that I could not work any odd hours that I wished, or I couldn't go in to lab over the weekends, or that I couldn't get on phone call or something with my colleague at 7 p.m. I could not wrap my head around the fact that I couldn't do it. And I kept trying to do that with a lot of disappointment, self-disappointment, because I couldn't do it. But I think understanding that my priorities have shifted and that that's okay was a major step towards it. So what I've done in kind of, this shifted sense of priority is surrounded myself with very, very, very accommodating, adjusting, and understanding people, including my mentor. Currently, my mentor is Simon John, and he is, one of the he's fantastic. Surround yourself with people who not only understand but also appreciate what you do in your position. And also surround yourself with people like Miruna, who I can't recount the number of times I've called Miruna and been like, oh my goodness, I can't do this, I don't know how to do this, and blah blah blah, blah. Like go on a complete rant about my inability to work outside of hours, or inability to work even period some days because my daughter is sick at home or something. But at the end of the day, it's okay because my priorities have shifted, and there is the work-life balance. There's no such thing as a work-life balance. I feel like the more you try to balance things, the more you get disappointed. I just now have assumed that what I do is good science, and that good science can come as part of me having a life, and not the other way around. I don't have a life as part of my work. I do my work as part of like the really, really good life that I enjoy as part of having a family and my daughter and all of that. And so I look forward to that, and I think having that perspective has really changed a lot of how I deal with science. And it's a fallacy that you cannot do good science if you also have a life outside of science. So yeah, surround yourself with people who are supportive and appreciative of that, and who also share the same goals and share the same sort of priorities towards life, I guess.
Yeah, Liz commented in the bottom, said it's amazing advice. I agree. That's all the questions that I had for right now. And we have a few more minutes left. So if anyone else wants to ask any last minute questions or drop it in the chat. If you'd like to add your contact information in the chat, we can definitely contact you after if we have questions later too.
If no one else has a question, I would ask, and something that I think links very much with what you said before, which again, like I think it's absolutely amazing and something for all of us to remember at different stages of our careers, and like whenever when we grow, to come back to this idea and always remember that priorities do change. I think you said it fantastically. I can thank you enough for that. So I think like the one last question that I think I would like to ask is, when did you feel that is the good time to have that addition into your life, and that is your daughter? And that is something that we all struggle with since we're in school. Like, do we want to go to school, or do we want to have a family? Do we want to start a PhD, or do we want to have a kid? And I think this will perpetuate it, and a lot of us can relate to this question. So what's your take on it?
A good time to have a family or not, or whatever, is the good time that you determine is a good time. Don't let anybody else tell you that it's not a good time. Like, I've had my fair share of people say, oh, if you have a kid in grad school, my God, you're doomed. Their career is doomed. It's not the case. I know my fair share of people who have had kids in grad school and who are thriving and doing wonderfully well. I've had my fair share of people also say, if you have a kid during your postdoc years, forget about academia. It's going to swallow you whole. That's not the case. I'm continuing on in academia. I'm doing well. I want to say I'm doing well. I hope I'm doing well in academia. And I want to see myself continue to do well and do good science in academia, irrespective of when I chose to have my family. I've also heard the answers of wait until you are, assistant professor to have a child. And the same number of people who say, oh my goodness, how are you going to build your own lab while you have your own child, because your lab is going to be your child. I've had advice saying, wait until you're tenured. But tenure, like the average age of tenure now is upwards of 40. So it kind of biologically becomes impossible at that point. Well, not impossible, improbable at that point. So I think advice, ranges all the way from, don't do it, to do it, to do it here and do it then, or like whatever. It doesn't matter. What matters is what's right for you, what's right for your family, and what's, how it adjusts, how you're able to adjust those priorities and move forward with whatever decision you make. I wouldn't listen to anybody who says otherwise, because your life is yours, and it's truly your timeline.
Thank you. I think that's something fantastic to even finish on. I think it's such a great take-home message. Thank you so much, Revathi. I knew it's going to be inspiring. It is always for me to talk to you as a friend, and I'm pretty sure for everyone else and for everyone who will check our video, because we do have people coming back to these interviews, and hopefully your stories will continue to inspire people that are part of our community. So thank you again for accepting our invitation. I think this was absolutely great.
Thank you very much. Thank you so much. I really appreciate you guys doing this. I think this, this needs to be, I think all universities need to have this. Hearing from women in science really inspired me when I was, struggling, and, there were a lot of aspects of my career that were under question. And I think what you guys are doing is just simply fantastic. So keep up the good work.
Thank you so much.
Thank you.
Thank you so much.
Thank you, everyone.
Thank you.
Bye.
Connect with you soon.
Bye.
Take care.
Thank you.